Good afternoon, student. Today we are going to talk about another topic in the syllabus, which is microbiology of soil, water, and air. Today we'll read about the microbiology of the soil, the microbes which are present in the soil, how they are behaving in the soil, what is their role in the soil, what kind of microbes are found in what kind of soil. So all these topics we'll cover about the soil microbiology today. And in the next class, we'll complete the topic about the water and air. This is our last topic for the syllabus. So let's read about the soil microbiology. Yeah, I'm, so microflora of soil, water, and air. And in this row, first lecture is microbiology of the soil. So learning goals of this lecture is basically we'll try to understand the different classification of schemes of the organisms which are found in the soil, how this classification is related to the organism's role in environment process and its vice versa, how the environment is playing role in the taxonomy of the organism or in the distribution of the organism. Right? So microbes are present in everywhere in every type of the soil. And if you see, this is like a very wet soil with the roots. A lot of microbes are present in the roots. And this is a diagram of one of the root nodules bacteria, rhizobium. So to understand the nature of the soil, it is necessary to know what kind of microorganisms are present in the soil. So that's a very important topic. Okay, and soil is a habitat of various kind of microorganisms. So if you see this slide, you can see that there are plant roots, there is water, there are soil particles, there is stone, and everywhere there are different kind of microorganisms which are present. So these are the different classification which is found in this diagram, and you can see that the number of bacterial colonies are present inside the root hairs, outside the root hairs, near to the rocks, near to the water and soil particles. So this shows that the soil is a habitat for number of bacteria and there are number of microbes, and these are present in different in different constitutions of the soil. For an example, a 1 gram of soil contains almost 100. This is almost like 1,000 lakhs. 10 crore bacterial soils are present, bacterial cells are present in 1 gram of soil, and almost 1100 species of bacteria are present. So when we read about the, if you remember about the culturable and cultural diversity, I told you that the cultural diversity, by cultural diversity, we can only get 1% of the micro communities which are present in the any environment. So 1% of this is 110 species of bacteria. So by culturable, we can get 110 species of bacteria. No, even we cannot get 100. We'll get maximum what we can get is like 10, 11 or 12. So the cultural diversity, when we try to understand the cultural diversity, we know that the number of the estimation of the bacterial species or the bacterial cells which are present in the soil is very underestimated, and then that's why we use unculturable techniques or with the help of next generation sequencing. And what we found out in that lecture, that with the help of next generation sequencing, we can estimate probably like almost 200, 300 or 400, 500 different species which are present in the soil. So we can come very close to the real number which are present in the soil or any environment. Okay, as well as there are fungi and larger animals which are also present in the soil. So microorganisms in the soil, they can be classified or they can be divided on the basis of ecosystem function, on the basis of their trophic physiological group, and on the basis of phylogenetic and taxonomic identification. So we have already read about the phylogenetic and the taxonomic identification when we read about the classification and the cultural and non-cultural diversities of the communities. So today what we'll do, we'll complete these two things, ecosystem function and trophic physiological group. Sorry, trophic physiological group. So first, ecosystem function. So on the basis of ecosystem function, yeah, on the basis of ecosystem, these microorganisms or all the organisms which are present in the soil can be divided on as producers, primary consumers, secondary consumers, tertiary consumers, or the decomposer. What are producers? Producers are those organisms which produce their own food and energy, and these are basically plants or the lower photosynthetic organism, like the cyanobacteria or the purple cerebral bacteria or other bacteria where the chlorophyll is present or the pigments are present. These produce food for all the producers. They produce food for themselves as well as for the primary consumers, secondary consumers, tertiary consumers, right? So what happens? This energy or the food moves from producers to primary consumer, who graze on producers, and then primary consumers are grazed by the secondary consumers, and secondary consumers are grazed by the tertiary consumers. And everything, whenever these organisms die, there are decomposers at the top of the food chain, where these decomposers, what they do, they use all these whenever this organism die, they decompose them back to the minerals which are present in the soil. So we can see that the organic materials are transferred from producer to the decomposers, and the inorganic materials or the carbon dioxide is transferred from decomposers to back to the soil. So on the basis of ecosystem function, these are the divisions. So what are producers? Producers, as I already told you, these are plant, green algae, cyanobacteria, mineral oxidizing bacteria. Yeah, one more point is mineral oxidizing bacteria or the lithotrophs. They also produce their own food, but with the help of chemical energy, not with the help of light energy. So those are mineral oxidizing bacteria. So do the inorganic energy production. Then the consumers. So consumers are feed directly on plants or on the other consumers. This include animals like macrofauna, moles, snakes, everything which is larger animals, mesofauna, ants, worms, and the microfauna like nematode and protozoa, and microflora like bacteria and so these are the consumers. And then the decomposers. Decomposers are microflora like the bacteria, including actinomycete, and fungi, including yeast. Here, one more point is there, that these are the told about the microfauna. There is also microflora, which are bacteria and fungi. So these are also included in the consumers. Some bacteria can function as consumers, and other bacteria, they work as the decomposers. So microflora, bacteria, and fungi. This groups also comes here. So macrofauna, mesofauna, microfauna, nematode and parasite, and then microflora, bacteria and fungi. Okay, fourth group here. And then in the decomposer, it is mostly microflora, which is bacteria and fungi. So on this was a classification on the basis of ecological parameters. Now the classification on the basis of trophic categories. So on the basis of trophic categories, this can be defined on the basis of source of carbon for cell synthesis and mechanism used to generate energy. Okay, so this can be divided on the basis of two. So first, the source of carbon for cell synthesis. So source of carbon for cell synthesis, if it is CO2 or inorganic compound, then those are autotrophs. And when this autotrophs converts CO2 to the organic carbon, it is used by the heterotrophs and again converted back to the CO2. So on the basis of carbon source, these can be divided in autotrophs and heterotrophs. Okay, so autotrophs are mainly producers and heterotrophs are consumers and decomposers. Okay, so this was a classification. Producers, consumers and decomposers was on the basis of ecological parameters, like on the basis of ecology. While on the basis of trophic, this could be producers are autotrophs and consumers and decomposers are heterotrophs. Got it? Then on the basis of energy generation, this is very important. On the basis of energy generation, organisms are divided in number of groups. What is the importance in this? That energy is harnessed during any energy is harnessed during the movement of electrons, or when it requires a reduced electron donor and an oxidized electron acceptors, right? So oxidized species, you already know in the chemistry that the oxidized compounds can accept the electron and reduced compounds can release the electron. So when a compound is reduced, it releases the electron, and which has taken over by the oxidized electron acceptor, and in this process the energy is released. And this is a dissimilatory reaction. Why we are calling energy production is a dissimilatory reaction? Because nothing assimilate in the body, right? Or the only thing which we get is energy. There's no assimilation. Like there's not a formation of a or a nucleic acid or the amino acid or any other living molecule. Okay, if those compounds are assimilated, if this electron acceptor or electron donor they are assimilated in the body system in the formation of some compound, then it becomes an assimilatory reaction. Dissimilatory reactions are those reactions where just the energy is produced. So energy is produced and the materials are released. So material, whatever electron donor and electron acceptors are used, they are released as oxidized electron donor. So reduced electron donor becomes an oxidized electron donor, and the oxidized electron acceptor will become a reduced electron acceptor. How this takes place, will come read in few more slides. So on the basis of energy generation, electron donor types are organotrophs, which use organic compound like the consumers and decomposers. And another is lithotrophs, which use inorganic compounds. They are producers, invariably use carbon dioxide for cell synthesis. And then the phototrophs, which use light. Producers invariably use carbon dioxide for cell synthesis. Got it? So these are the three types. Organotrophs, which use organic compound for the cell synthesis, energy generation, and these are consumers and decomposers. They use organic compounds for the cell synthesis and energy. Similarly, lithotrophs, these are producers again. We have read about them, and they use inorganic compounds and as well as CO2 for the cell synthesis. Okay, so inorganic compound and the CO2 for energy generation and cell synthesis. Third are phototrophs. They use light for energy and CO2 for the cell synthesis. Light and energy for cell synthesis, and light and CO2 for cell synthesis and energy. So we can call organotrophs, organoheterotrophs as heterotrophs, photoautotrophs as phototrophs, and lithoautotrophs as autotrophs. Okay, or we also call phototrophs as autotrophs, autotrophs as phototrophs. So these are generally terms which are used as a synonyms. We should understand autotrophs are the one who make their own food, who use light energy or the chemical energy for preparation of their own food. So when they use light energy, these are phototrophs. When they use chemical energy, these are lithotrophs and both of them together are known as autotrophs, while the organoheterotrophs are the heterotrophs, which use organic compound for carbon and energy. All right. So energy generation category. So is first one energy generation category we read here was on the basis of electron donor type. So what are the electron donors? So when the electron donors are organic compound, these are organotrophs or the organoheterotrophs. When the electron donor are inorganic compounds, then these are lithotrophs. When the electron donor is light or CO2, then light, then it is phototrophs. Right, so these are the categories we just read. Now energy generation categories on the basis of electron acceptor types. So on the basis of electron acceptor, these are the three different types of electron acceptors which could be there. First is oxygen, which is an electron acceptor in all the aerobic species in the world. And then these are oxidized forms of nitrogen, sulfur, and metals, as well as halogenated organic compounds. So these are the different electron acceptors which can be used, right? So on the basis of electron acceptors, we can divide them as aerobes, aerobic aerobic microbes, or aerobic species which use only the oxygen, which can only use oxygen as a terminal electron acceptor. Then there are facultative anaerobes. Facultative anaerobes are that the generally which are aerobic organisms, but can also grow on anaerobic electron acceptors or the other electron acceptors other than oxygen. And anaerobes are the microbes or are the bacteria which grow only on on the electron acceptors other than oxygen, because oxygen is basically poisonous for them or toxic for them. So aerobes is obligate or strict. We know that all the organisms, including higher animals, macro, meso, and microfauna, all of these are aerobes. And microflora, mostly fungi and many bacteria, including all actinomycetes, are aerobic. Facultative anaerobes use oxygen if available. If oxygen is absent, they can use the alternate electron acceptors like the nitrate, iron, manganese, and sulfate. So all of these are alternate electron acceptors. The groups which are included here is bacteria and eukaryotes. Eukaryotes, yeah, but they are capable of limited fermentation, and which I've just told you here eukaryotes to just give you an idea that there's a fermentation which takes place in many eukaryotes, but that fermentation is mostly again done by the microbes which are present in the eukaryotes. So that is not very clear, right? So if somebody says that yeah, eukaryotes are facultative anaerobes, yeah, for little time or for some time, but not exactly facultative anaerobes, because they cannot live very long for the without oxygen. And bacteria, yeah, there are a number of bacteria which can live as facultative anaerobes. Then strict anaerobes, unable to use oxygen and electron acceptors. So what they use is oxidized metals, CO2, halogenated organics, as well as other oxygenated compounds like the nitrate, iron, manganese sulfate, or these are alternate electron acceptors. Lack protection against the toxic oxygen species. I've already told you last time that anaerobes, why they cannot use oxygen, because they don't have the enzymes which can detoxify the reactive oxygen species like the peroxide, superoxide, and for these the enzymes which are required is peroxidase, catalase, and superoxide dismutase. All these enzymes are not present in anaerobes, and that's why during the process when the electron is accepted by the oxygen, terminal electron as a terminal electron acceptor, these number of reactive oxygen species are formed like the peroxides and superoxide oxides, and these peroxide and superoxides can only be converted into a stable oxygen form with the help of enzymes like superoxide dismutase, peroxidase, and catalase. All of these are different function, slightly different function. And with the help of these enzymes, the reactive oxygen species is changed into a stable oxygen species or the water. And this cannot be done without these enzymes. And all these enzymes are not present in anaerobes. So the example for this is bacteria and archaea. So as we know that the redox reactions and are the coupled reactions that we need a reduced electron donor and the oxidized electron acceptor. So these are basically the redox couple of one reduced compound and the another is oxidized compound. Both of these are required for doing this redox reactions. Okay. And yeah, so these are, suppose this is an electron donor, which is like a CS2O, which is like a form of a glucose or the carbohydrates, which is converted into CO2, electron donor. And in this process, whatever electrons are coming out has been taken out by oxygen, which is converted into water. And this is the phenomena which takes place. So all the electrons which are coming out in the process of converting glucose to CO2 in the process of respiration, and this electrons goes to terminally to the oxygen, which is converted into water. And this is generally happens at the 700 millivolt volt, and this condition is known as oxic condition, which takes place in most of the aerobic organisms. Then if oxygen is not present, then there are certain organisms which can use nitrate, sulfate, iron. So when in these conditions, when the electron donor, which is like glucose, again converted into CO2, and the terminal electron is taken up by nitrate and converted into nitrogen, or taken up by ferric, Fe plus 3, and converted into ferrous, Fe plus 2, right? So this is like a suboxic conditions, or number of facultative anaerobes do these conditions, and this is at the happens when the redox potential is 400 millivolt. So this happens at when the redox potential is 700 millivolt. This happens generally, reaction happen at when the redox potential is 400 millivolt. And the third one is again lower than this, when the glucose is converted into CO2, and all of these are not present. Then the sulfate can act as a terminal electron acceptor and convert it into hydrogen sulfide. And this reactions happen at a very low redox potential, which is like 100 or minus 70, actually minus 70 millivolt. And this is an anoxic condition. This is totally anoxic condition. So this is on the basis of one electron donor, which is glucose, and different electron acceptors, right? So in the aerobic organism, this reaction is taking place when the electron acceptor is oxygen. Electron donors can be different. Most important is organic carbon, which we see here, glucose, or the carbohydrate, and other food source or other compounds can be ammonium, ferrous ion, or hydrogen sulfide. All these three can also act as a food source, or the reduced electron donor, and the electron acceptor here is the oxygen. Then all of these total reactions organisms are known as aerobic organisms. This is well known. We all do it. Organic carbon, we use carbohydrate, and oxygen is used as a electron acceptor. Then there are some ammonium oxidizing bacteria which use this reaction. Then there are some iron oxidizing bacteria which do this reaction, like the iron oxidizing bacteria like geobacter. And then there's another group of bacteria which do the sulfide oxidation, and sulfide oxidation like a thiobacillus or the archobacter. These are the organisms which convert hydrogen sulfide into again to sulfur, back to sulfur, and the electrons are taken up by electron acceptor, which is molecular oxygen. So you understand, here we just read the example of one electron donor which is carbohydrate, and the electron acceptor were different. If this is an electron acceptor, just the oxygen is electron acceptor, and there could be different electron donors like the organic carbon, ammonia, ferrous ion, and hydrogen sulfide. All of these organisms are known as aerobic organisms, right? So here it is shown, different electron donors. Electron acceptor is only oxygen. When the carbohydrate is used as electron donor, the energy which is released is very high. This is like 400 kilojoule per mole. The negative sign shows that the energy is released, exothermic reaction, right? And then another one is ammonia, which is when it is converted into nitrite. Then the third one is ferrous ion converted into ferric in the presence of oxygen. And the fourth one is S2S is converted into sulfur, not to sulfate, but like, yeah, any oxidized form. Sulfide is a most reduced form of sulfur. So this reduced form convert into any oxidized form like sulfur or sulfate in the presence of oxygen, and then it is the energy which is released is very low, like this is like 36 kilojoule per mole. So very less energy. So this, and you understand, these organisms are use different electron donors, but the energy these reactions are not very favorable. This is like a, you can see this is what, this is not phototroph, this is the, sorry, this is, these are like um, because these are not using organic carbon, so all these kind of bacteria are what, these are lithotrophs, right, using chemical, using inorganic compounds for the production of energy, right? So ammonium oxidizing, iron oxidizing, and the sulfide oxidizing. Then another one are anaerobic organisms. So in these anaerobic organisms, food source can be anything. Most important is organic carbon, as I told you. Other ammonium, ferrous, and hydrogen sulfide can also be the electron, can be the food source. And electron acceptors are not the oxygen, because this is anaerobic organism, but nitrate, magnesium, ferric ion, or sulfate. So you understand my point? Here there's a difference. Here oxygen is not an electron acceptor. Here the electron acceptors are anything other than oxygen. And food source is all these food sources. Most important one is inorganic carbon, and other three are there, right? So if we see it here, so electron donor is carbohydrate, and these different electron acceptors, right? So we are not talking about oxygen here, because we are talking anaerobic, only anaerobic. We already know that when the electron donor is carbohydrate and when the electron acceptor is oxygen, the energy release is 400 kilojoule per mole, right? Similarly, 400 kilojoule per mole. Similarly, when when nitrate is converted into ammonia, ferric is converted into ferrous, and sulfate is converted into sulfide. Right? And the energy release is very low, which is in the presence of carbohydrate. This is inorganic, this anaerobic reaction. And you understand my point? These are right now, these are different electron acceptors, right? Different electron acceptors, not in the present. So these are different electron acceptors this time, right? So different electron acceptors. In this different electron acceptor, oxygen is this one, which we don't want to use right now, because we are studying about the anaerobic organism. Then comes the nitrate, which is very close to oxygen. You can see that energy releases between the 436, means less than the oxygen, the energy which is released by reduction of oxygen, but more than the sulfate. Okay, and then it is ferric, which is converted into ferrous. And then last one is when sulfate is converted into sulfide in the presence of electron donor, which is carbohydrates. So carbohydrates is converted into CO2, and when the electron acceptor is sulfate, the energy release is very low, which is 36 kilojoule per mole. Right? This is the same kind of thing which we read here, right? When sulfide is converted into sulfate, when we, in the last slide, when we read that the sulfide is converted into sulfate in the presence of oxygen. So that was a aerobic reaction, right? Because of all the electron acceptor, what we were reading in the last slide, if we repeat, wait. This one, if we repeat, so you see that the sulfide is converted into sulfate in the presence of oxygen. So aerobic reaction, and then the energy release was very low, which is 36 kilojoule per mole. Similar amount of energy is released when sulfate is used as electron acceptor. Sulfate is used as electron acceptor, converted into sulfide, and the electron donor there is carbohydrate only. So which we write in the next slide. Here, when the carbohydrate is used as an electron donor and the sulfate is used as an electron acceptor, it's the same amount of energy, which is 36 kilojoule per mole, was released. So you understand the difference of different electron acceptor and electron donor, right? So So these are just the revision. These are the primary producers. Then these are the consumers, secondary and tertiary soil ecosystem. We call them as primary producers, and then the primary. These are the. Sorry, no. What I mean to say is. Um, yeah. Yeah, so here what is there, that these are basically about the bacteria and fungi which feed on root deposition, plant debris, plant root, and so these are like the primary consumers, and then the primary consumers are are fed on, and so that then the secondary consumers are fed on primary consumers, and the tertiary consumers are fed on secondary consumers. So this is like the ecology, soil organisms and ecology of these soil organisms, right? So just to repeat everything what we have read, autotrophs and heterotrophs. In the autotrophs, there are two types of producer, phototrophs and the lithotrophs. So phototrophs can be aerobic bacteria, can be aerobic, which are bacteria and eukarya, most of the eukaryotes, and the facultative anaerobes, bacteria and anaerobic bacteria. In the lithotrophs, we have aerobic bacteria and archaea, facultative anaerobes, bacteria, anaerobic bacteria and archaea. Then comes the heterotrophs. In this, they are consumers and the decomposers. Consumers, organotrophs, which are aerobic, mostly eukarya. Okay, as well as there are bacteria also, aerobic bacteria. Okay, and then comes the decomposer, organotrophs, which are aerobic, bacteria, eukarya, facultative anaerobes, which could be bacteria, eukarya, and archaea. I told you about the decomposers. We are generally not we are not very effective facultative anaerobes, but can do a bit of limited fermentation. So that's why we come here as facultative anaerobes, like some of the eukaryotic organisms. Here V means like some of the eukaryotic organisms. So facultative anaerobes, and then the anaerobic organism, anaerobic decomposers, which are bacteria and archaea. So this is all what we have read till now, except that energy part, energy generation, division on the basis of carbon used for energy, or the electron donor on the basis of electron donor, and or on the basis of electron acceptors. Then comes the decomposers, right? So decomposers are obtain their carbon from producers or from the consumers, which are dead organisms, dead biomass, excretion and secretion. So passively they live on dead biomass, excretion and the secretion of the different organisms. So those are known as saprotrophs. Okay, so live on dead or biomass, excretion and secretion. Those organisms are known as saprotrophs. Actively on living biomass, those are necrotrophs, attack and decay biomass. Necrotrophs, like many pathogens, and biotrophs obtain carbon from plant in exchange of microbe supplied nutrients. So those are symbiotic organisms like the rhizobium. They obtain carbon from plant in exchange of microbe supplied nutrient like the rhizobium. So there are three definitions: saprotrophs, living on dead molecular organisms; necrotrophs, which attack and decay biomass like the pathogens; and then the biotrophs, which are symbiotic. So example of saprotrophs, the saprotrophs, the organic material which is used by saprotrophs are plant litter, plant exudates like the root exudates. Exudates are root particles, animals, bodies, excretions, and microbial cells, exudates. Okay, all of these are the organic material which can be used by saprotrophs. Necrotrophs, they attack on living plants, so attack and decompose living plant part or the animal parts of plant pathogens like fungi. Example, Pythium and Bacteria irvania. Then biotrophs, you all know Rhizobium and Actinorrhiza, all are the fungi. There are number of fungi which can help in phosphate uptake, known as mycorrhizal fungi. These all are biotrophs, right? Then on the basis of organic pool of carbon in soil, uh, organic carbon in soil, we can divide this in two different types of thing. Just all right. So here there's a classification according to a growth of characteristic. So two general pools of organic carbon. One is resistant to degradation and another is liable to degradation. So organic carbon, which is used by the decomposers, this can be divided in two types. One which is resistant to degradation, means which is not easily degradable, and another is liable to degradation, which can be easily degraded. So example for this is, for the resistant one is soil organic material like the humic material, humus, humic minerals, amount are relatively constant in the environment. And their amount in the environment is relatively constant throughout the year with the time and everything, right? While the liable to degradation, which can be easily degraded, fresh inputs like the root exudates, soluble compounds released from dead cells, amount are highly variable, and it is variable with the help of, especially and temporarily, means with the help of, with the seasons and with the space, they change variable, they are very variable. Okay, so soil organism may adapt to exploit one or the other of these pools. So soil organisms, some of these organisms can use this one, which are very easy to degrade, and other kind of microorganism can adapt themselves to use the organism, the matter which is hard to degrade. There are two definitions which was given by Vinogradski in 1900. One was zymogenous and another was orthocanthonos. So zymogenous, what are zymogenous? Actually the zymogenous literally means ferment producers. So the organism which can attack readily available carbon sources are zymogenous. So organisms which use which are decomposers, which can use the easily decomposed organism, easily decomposable material, are known as zymogenous. Orthocanthus, on the other hand, are self-feeders. The name means self-feeders of the earth, or humus degraders. Means they attack the material which is resistant. Okay, and they use a material which is resistant or hard to degrade material. Population densities of these zymogenous, they fluctuate widely. Why? Because when this easily available carbon is present, they grow very fast. They started growing very fast, and their rate of respiration or rate of growth is very high. However, rate of growth is very high. However, Athocanthus, they are actually the slow one. They attack the resistant material. Their population densities are relatively stable throughout the time because they grow slowly. Okay, and these are larger in size, 1 to 3 micron diameter spore forming bacilli, and these are smaller in size, 1 to 1.5 micron diameter or cocci. Right? There are another terms which is oligotrophs and copiotrophs. So copiotrophs means eutrophs or abundant feeders, means the organisms which use the easily available carbon source or easily decomposable or easily degradable carbon source are copiotrophs. On the other hand, oligotrophs are those which are actively adapted to the presence of low amount of readily degradable carbon sources, right? So these are oligotrophs are those ones which can adapt themselves to a low amount of readily degradable carbon sources or compound not readily degradable. So oligotrophs are the compound which can adapt themselves to the non-degradable compounds, not easily degradable compound, or even at the low concentration of readily degradable compound. So basically, oligotrophs are the one which are ortho, canthonos, and copiotrophs are mostly which we call zymogenous. So characteristic of oligotrophs. Okay, uh, they can be aerobic, but they lack the enzyme like the catalase and the superoxide dismutase, so they don't have a protection against the toxic oxygen species. Okay, they don't have full protection against the toxic oxygen species like the facultative anaerobes. High nutrient uptake capacity, high surface to volume ratio. Okay, so they have a high surface area, low volume, and high density of transporter sites. Okay, because they can grow they can adapt themselves to a very low nutrient conditions. So which means they need a high amount of transporter, highly efficient cells. High number of transporters should be higher, so that what? So that all the lower nutrient which is present in the environment can be taken up by the cell with the help of transporters. One more important point is that they are efficient nutrient management to allow growth. Means they for the formation of reserve polymer. So they also, whenever there is a higher concentration or little bit higher concentration, they what they do, they make poly beta hydroxy butyrate. Okay, so beta hydroxy butyrate, we call it as a PHB, polyhydroxybutyrate, PHB. And this PHB or PHA or PHB, polyhydroxyalkanoid or polyhydroxybutyrate. These are the organic compounds which can be utilized when there is a starvation conditions. So biosynthetic regulation. This polyhydroxybutyrate are also used as bioplastics nowadays. This is a polyhydroxybutyrate is researched as a bioplastic alternative to the chemical plastics or the polyethylenes. Okay, one of the example of the oligotrophs is arthrovector. Why? First of all, it has a cocci cells under low nutrient conditions, means the high surface to volume ratio, and rod shaped lower surface to volume ratio under high nutrient level. Okay. Uptake system favors organic acids, amino acids, reduces competition for sugars. Endogen, right? So this means the uptake system can also use organic acids or the amino acid instead of sugar as an electron donor. Endogenous metabolism of starving cell, 10% of that starving copiotrophs. Means endogenous metabolism means that there are, as I told you, there is polyhydroxyalkanoid which are present in the cells, which can be utilized for the metabolism. Cell are resistant to desiccation. Very important. Means even these cells does not form any spores, but still at the at the low water conditions or when in the very hot condition, these cells can have a resistance to desiccation. So all these point shows that the orthobacter is one of the soil oligotrophs. This is a morphology transition of orthobacter. You can see this is like a cocci cells in the low nutrient as the nutrient condition is increasing, the cells is becoming rod shape, and as soon as the nutrient is depleted, it becomes again the cocci shaped, right? So this is as the nutrient condition is increasing, the cells is becoming rod, and then in the nutrient starvation condition, they become the Kokai cells. There are two more terms which we should know. One is R strategist and another is K strategist, or R communities and the K communities. What does this mean? So this is a mathematical model developed for the same type of microbial communities, one which can use the easily degradable compound, or which can grow in the high concentration of the food, or high concentration of the resources, and another are the communities which grow in the low concentration of the resources. So this is a mathematical model for developed for them. Here, if we call it this R is a actually this should be the capital R. Yeah. Okay. R and K strategists, right? So yeah. So R, small r is a rate of population increase, the rate at which the population is increasing, and the K here is carrying capacity of the environment, means how much density of the organism can be supported by that can be supported in the environment. This should be 1 minute. Right. Okay. Yeah. So yeah. So K strategist, or the K, capital K. So K communities, you can also call them as K communities. K strategists are those which are optimized to function at high population sizes but low growth rate. Okay. So optimized to function at high population sizes but low growth rate. Okay. Means the high K, this is small k, and low growth rate, high K and low growth rate. And R selected are optimized to function at low population sizes, low K, but high growth rates. So if we see here, so R strategies or R communities are those where what we read was R value is high, but means the growth rate is high, but the population density is low. K strategies is population growth rate is low, but the population density is high. So if this is a rate of growth of population, and if this is a population density, which is inverse of basically resource. If the population density is increasing this way, the resource are decreasing this way, right? Population density is increasing and the resources are decreasing. Resource availability is decreasing. So here in this, R is where the R value, where the growth of population is, where the growth rate is high, population growth rate is high, but the total population is lower. So population density is lower here, and the growth rate is high. So till this point, these are the R communities. And when the population density becomes the higher, and the rate of growth has become lower, then these are the K communities. So R communities, you can understand that R communities favors what? When the resources are high relative to population. When the resources are high relative to population, then the R communities grow. They grow very fast. They grow, population growth is very high. They grow, start growing using all the resources. So they don't convert it into the population. They do a lot of metabolism. They convert, the growth rate is high. Growth rate will be when high, population growth rate is high when the there is a resource high, and these organisms started using them, right? When what happened when the resources deplete? Okay, at that time K strategies come because the growth rate is lower, okay, and the resources are lower, and at this point the K strategies, the K communities come over. So competitive crossover density is here, where the R and K communities both live together. But as soon as this condition comes and the resource ability decrease, the K communities favors can grow. So K communities can grow when the resources are low relative to population, and R strategies favor when the growth when the resources are high. So in turn, we can say that the Zymos genus, which use easily degradable compound, are also equivalent to copiotrophs and are also R strategists or R communities. Whereas cathonos are the communities which are oligotrophs, grow in the low organic load, low organic compound, low resources, and also known as K strategies or the K communities. Got it? So this is all about the soil microbiology. Now there's one more slide here which shows different kind of organic compounds which can be used by the different organism, like the primary producers, chemoproducer, mixotroph, consumers, decomposer, and number of examples of these bacteria. I would like to tell you that you actually should what to do. I'll send you this table in another Word file so that it is visible. It is easily visible. And what I want you to do is you should remember at least one example of each of this kind of community, right? Like in the obligate aerobes, if you see here in the photosynthetic, there are examples are given, okay, which can use oxygen. And then there are facultative anaerobes, right, which can use the anaerobic, grow in anaerobic conditions. And then there are examples are given, right? And then there are obligate anaerobes, and then the examples are given. Then in the obligate aerobes, then there are a number of examples are given for each type. So what I want you to do is you should remember at least one example of any bacteria for in each of these things. This will give you a more idea about that how this bacteria functions and how these bacteria are different from each other, right? All right, so this is the table which I'll send you also in the Word file, as well as I'm sending you this PPT so that you can study. There's one more thing which I want to tell you, right? This one. So in last, what can we infer from elements redox transition in biological chemical cycles about the micro driving forces underlying the individual transformation, right? So if let's try to think about that how these different transformations which we read about or aerobic compound, about the producers, consumers, primary consumer, tertiary and decomposer, how all these micro driving forces can what do can function in the biochemical chemical cycles, and as well as what can we infer about the elements redox transitions in biochemical cycles, and how these elements which are present in any environment, how these redox couples can influence the biochemical cycle. Let's try to think about it, that one environment which is an anaerobic environment, what kind of communities will be grow there? That will depend on number of things, as I told you. That will depend on what all compounds which are present there, and what all microbial communities are present there, what all transformations will take place, what do microbes get out of the reactions, okay, all those redox reactions, from the redox reaction, what is microbes getting? Are these dissimilatory reactions or the assimilatory reactions? How they are, these microbes get adapted to this their environment? What ecological trophic category of would mediate such a reactions? And which ecological or trophic categories, like the consumers, like producers, consumers, heterotrophs, organotrophs, or the ecological, so the ecological like the producers, consumers, trophic level like the autotrophs, heterotrophs, and are on the basis of energy, aerobic, anaerobic, what all kind of communities are driving these biochemical cycles. So think about one of the biogeochemical cycles, nitrogen cycle or the sulfur cycle or the carbon cycles. Okay, just take that cycle, take the diagram of that cycle, any biochemical cycle. Try to list what all microbes will be growing at what part in that cycle. Okay, try to figure out on the basis of this whole lecture, try to figure out which microbes will be growing at what places. Like for example, in the sulfur cycle, if there is S2S which is coming at the end product, what microbes will be growing when S2S is present and the nitrate is present. So S2S probably will be used as a electron donor and nitrate is used as the electron acceptor, and then what kind of community will be growing there. So take that one or two of those cycles and try to do a lot of this calculations with the what kind of microbes will be growing, and then the example of each microbes from this table. Okay, so try to do that. I'll give you one exercise also about that as an assignment, but right now this is just food for so you're thinking. So food for your thought. So read this presentation again and again. I'll send you this presentation as well as this video recording, and I hope you like this lecture of today. And our next lecture will be about the water and air microbiology, or the microbes which are present in the air and water. And that will be the last lecture for this syllabus, and then we'll have some interaction classes. Thank you very much.